Life Sciences Video Lecture 6, Fluids of the Body. All right, class, let's take a look at what we're doing in this lecture today. We are going to differentiate intracellular and extracellular fluid and name the three types and locations of extracellular fluid, and we'll name the barrier between each fluid. And we will define electrolyte and list 4 functions of electrolytes in the body and give examples of those, and 3 ways in which the body maintains acid-base balance. So, in general, what that means is we are looking at inorganic chemistry in the body. So, we had talked before about how organic versus inorganic chemistry, or organic versus inorganic molecules. The organic molecules are the large carbon-based molecules, and those are the ones that only show up in living systems. Inorganic molecules are small and not carbon-based, and they show up in living systems in that they create the environment of within which everything happens in living systems, but they also show up outside of living systems. So what we're looking at here on this slide are a few of the most common elements that we find in the body, and all of these guys would, and the molecules that they make, or the ions that they that they turn themselves into, are some of the most common ones that we find in the body. Sodium being one of the most common, and potassium. Those two are way up there, but calcium and magnesium and chloride also really high up there, and manganese as well. So, and there are others, but anyway, this is a little depiction of some of the atoms, some of the elements that we're going to be looking at in today's lecture. So, okay, let's take a look at the notes and the slides. So, basically, when we're talking about the inorganic chemistry of the body, what that means is we're looking at the environment of the body, the internal environment of the body. So that both means the environment that the cells live within. So what is it outside the cells that that is creating the environment that the cells live in? But it also means what's creating the environment inside the cell. So the internal environment of cells also. So both inside and outside cells, we find fluid. And so the fluid of the body is essentially the internal environment of the body. Okay, so something is not considered to be inside the body until it crosses a membrane, some cell membrane, and enters into fluids of the body, whether those fluids are in cells or outside of cells. Once something is in the fluids of the body, then it's in the body, it's inside. So so that creates the internal environment of the body. So as we know, the body is made up of about 2/3 fluids. So on this slide, we're looking at actually a slight distinction between male and female total body mass. And so they're showing on the female 45% solids, and on the male 40% solids. So that puts the female at 55% fluids and the male at 60% fluids, which is kind of interesting because we think of women as, you know, being more creatures of yin and blood in TCM terms. So we might expect more fluids, but if you break down the fluids, in comparison to solids in the body, we have a little higher percentage in males. So if we take that roughly 60% fluids, so 55 to 60% in the average adult, that percentage of fluids, if we take those fluids and break it down and take a look at what's what, we find all the individual types of fluids that we have in the body. So we can divide bodies into 2 main categories: intracellular fluid and extracellular fluid. So on the chart here, next to the man's fluids, we see 2/3 intracellular fluid and 1/3 extracellular fluid. So intracellular fluid, also abbreviated ICF, intra means inside, so intracellular means the fluid inside of cells. So 2/3 of our 2/3 of water that makes up our body is found inside our cells. The remaining 1/3 is found outside cells. So that so extracellular fluid, extra means outside, and extracellular means outside of cells. So 2/3 of our fluid is found inside cells, 1/3 of our fluid is found outside cells. So of the fluid that is outside cells, depending on where it is going to have a different name. And depending on where it is, it's going to have a different function and a different, it creates a different environment. It's a different part of a different aspect of the internal environment of the body. So we have 3 major divisions, although we could come up with at least one other here, but 3 major divisions of that extracellular fluid. So extracellular fluid can be, so 80% of that is what we call interstitial fluid. So interstitial means between the cells or within the tissue. So between the cells. So in that picture on the right there, we see cells and we see just sort of the blue background color. That's fluid that's in between the cells. So that's our interstitial fluid. So 80% of the fluid that is not inside cells is immediately right outside cells, making up that interstitial fluid. And then on the chart they show, what is it? Can I zoom in? Oh, yes, I can. 20%. So that remaining 20% they're just showing as plasma. But we really could break that remaining 20% down and be a little more specific about it. So 20% of that is found in the plasma. Some percentage, or 20% or so, some percentage of it is also going to be found in the lymph, and a very small percentage, maybe 1% or so, is found in the cerebral spinal fluid. So each of those places, the lymph, the blood, the cerebral spinal fluid, or the lymph, the plasma, I should say, in the cerebral spinal fluid, those are all extracellular fluids, just like interstitial fluid is, but those are fluids that are contained within a vessel or within a space. And so it is their, the characteristic of being inside that, inside a blood vessel that makes that fluid blood plasma. And if that very same fluid went from blood to lymph, we would now call that, from a blood vessel to the inside of a lymph vessel, then we would call that same fluid lymph. So lymph is the fluid of the fluid that flows within the lymph vessels of the lymph system. Plasma is the fluid that flows in the blood, is the fluid aspect of the blood. So in the blood we have red blood cells, white blood cells, and platelets. Those are the cells of the blood. And then plasma is the fluid that they float around in and get transported, and other things are dissolved and not dissolved, just suspended in the fluid of plasma. So. And then the cerebral spinal fluid also, that's fluid that surrounds the brain and spinal cord, so it's contained within the meninges of the central nervous system, and that's the only place it's found. But we can take a droplet of that water, put it in the blood, and boom, now it's plasma. So it's simply the location of each of those fluids that designates them as being lymph or plasma or cerebral spinal fluid or interstitial fluid. As far as all of the extracellular fluids go, because there's always exchange, right? So nutrients are going from the blood, so they might be suspended or dissolved in the plasma, and then they might leave the blood with leave the blood with some fluid that leaves the blood and go into the interstitial space. And so then it's interstitial fluid, and that interstitial fluid then may leave that interstitial space and go inside a cell. And so once it's inside a cell, then we're going to consider it intracellular fluid instead of extracellular fluid. But you can see that these fluids are always changing location, changing position. And so each of these locations, though, needs to be designated and kept separate from the other locations, because different processes need to happen in each of these bodies of fluid. And so we have to have something that designates one body of fluid from another. And basically what that is cell membranes. So cell membranes are a fascinating topic in and of themselves, and we will take a class to talk just about those, or at least a lecture to just talk about those. But they're highly structured, and they display an amazing amount of intelligence as far as subtly manipulating or changing their permeability to allow different things in or to not allow different things in and things like that. So, but basically when it comes down to it, it is cell membranes that designate each of the fluid areas in the body. So intracellular fluid, also called cytosol, that's the other term for intracellular fluid, or that fluid inside cells, that's completely contained inside the cell membrane. So inside the cell membrane we have cytosol, or intracellular fluid. Outside the cell membrane we have we have interstitial fluid, which is one of the types of extracellular fluid, and those two are separated from each other by that cell membrane. Inside blood vessels are made out of what? They're made out of cells. And so it's those cell membranes that make up the blood vessels that separate the fluid inside the blood from, it's actually the first layer of separation is the fluid inside the blood, so the plasma, separating that from the fluid inside the cells, the cytosol of the cells that make up the wall of the capillary. And then if we go through another layer of membrane, now we're back into that interstitial space, and we have and it's interstitial fluid. So you can see that cell membranes divide each of these areas of fluids. So same thing with the lymph vessels, same thing with the cerebral spinal fluid. These are compartments that are created, that are built and maintained by cells, or separated from one area to another by cells, and so it's those cell membranes that create those distinctions from one area to another. So the fluids of the body make up the internal environment of the body, and the pH and the presence of various molecules and ions in those fluids help those fluids to support the processes that need to happen in them. So what we're looking at here is a chart that shows some of the most commonly occurring ions in the body. So ions, remember, can be atoms, so you can have a single atom acting as an ion, or you can have a small molecule acting as an ion. And what we have here are some of the more common ions that we have in the body. On the left of the chart is sodium, and so for each of these, then we see how much of each of these substances are found in the three different key fluids that we have in the body. So for instance, for sodium, you can see that the red bar is way up there at like 150. So we have so let's see, what is that? Micro something per liter. I actually do not know what that denomination is, but we can at least see the relative different levels here. So we have a lot of sodium in the blood, right? That's not surprising. Blood tastes salty if you ever get a taste of blood in your mouth, and we have a lot of a lot of sodium in interstitial fluids, but much less sodium in intracellular fluids. So inside cells there's not as much sodium, but if we compare that with potassium, we almost have like the complete opposite. We have only a little bit of potassium in the blood plasma and in the interstitial fluid, but lots of potassium in the intracellular fluid. So, and so we can look at each of those different ions and kind of get a sense that that every fluid in the body is not the same as every other fluid in the body. They vary considerably with the levels of different ions that are in it. And different ions in the fluids will mean a different pH and different reactions that could be happening within each of those fluids. So in the body, these, so like the things we're looking at in this chart, if we zoom in on them, we have sodium and potassium and calcium and magnesium, and then we have something called bicarbonate and chloride ions and sodium phosphate and sulfate. Those are the things that we're looking at here. And so all of those things make up what we call the electrolytes in the body. So the electrolytes are the ions in the body. And so again, ions can be single atoms or ions can be small molecules, but ions by definition have a charge. So they're either positively charged or negatively charged. The ions in the body have, or I could say the electrolytes in the body, all have very specific functions that they serve for us. So let's take a look at some of those things. Let's see. Let me just look at the notes and make sure I said everything I need to. Electrolytes in body fluids. We have different levels of electrolytes in fluids. There's a list in your notes of these same electrolytes that we're looking at on the chart here. A very similar chart to this is in your book on page 1043. Okay, so what we're going to look at here are the 4 functions of electrolytes in the body. So why do we have electrolytes? Why is it so important that, number 1, we have electrolytes at all in the fluids of our body, and number 2, why is it so important that we keep them at specific distinct levels within the different fluids, and why are the different fluids different from each other? So here we go. Functions of electrolytes in the body. The first function of electrolytes is to control osmosis. And okay, so the beaker on the left has a 10% sodium chloride solution. That means it's 10%, it's salt water that is made up of 10% salt in a 20% solution on the right side. And so, but this is all within the beaker on the left there. And then there's a membrane between those two sides that is permeable only to water. So what that means is that, so basically, the bottom line with osmosis is that water always tries to even the playing field, in a sense. Water always tries to make it so that things are evenly distributed within it. And so here we have this uneven distribution of the salt that's dissolved in the water. There's more salt dissolved on the right side of the beaker than there is on the left side of the beaker. So osmosis will happen in response. And so over time, we look now at the beaker on the right to see what happens after time passes. And what happens is that water passes from the left side of the beaker to the right side of the beaker. So it's passing, and it will keep passing through that membrane until it evens out the amount of sodium chloride dissolved in the water on both sides of the membrane. That's osmosis, the movement of fluids, and that's how electrolytes move fluids, is because fluids have a tendency to want to even out the distribution. And so if electrolytes are more concentrated on one side of a membrane, so in the body that would be, say electrolytes are more concentrated inside a cell or more concentrated outside the cell, fluids will tend to want to cross that membrane to even out the distribution of those electrolytes. Now cell membranes are smarter than this membrane that's in this beaker. And so even though water wants to cross it and even out the distribution of the electrolytes, cell membranes have the power to say yay or nay about that, because sometimes we want different concentrations. Well, all the time we actually want different concentrations of different things on different sides of the membranes. So, but this is the tendency for water to flow like this, and membranes have the ability to allow or not allow that tendency to happen. But this is osmosis, and we can control osmosis. In other words, control the movement of water across a membrane by controlling the amount of electrolytes that we have on one side or the other of the membrane. So we use electrolytes to control osmosis. That is number 1. Number 2, second function of electrolytes in the body is to create what are called buffer systems. And so, like I mentioned before, all of our fluids in the body need to be maintained at the proper pH. And so what we're looking at on this chart are the buffer systems of the body. So we have, at the top of the chart, it says buffer systems. Below that, we have 2 categories of fluids. So on the left, we have ICF, so intracellular fluid, and on the right we have ECF, so extracellular fluid. So, and then across the middle we have the 3 major buffer systems, and then where they occur. So in the intracellular fluid, also known as cytosol, we use the phosphate buffer system often. Both in the intracellular fluid and in extracellular fluid, we use what's called the protein buffer system, and then pretty exclusively in the extracellular fluid, we have the carbonic acid bicarbonate buffer system. So buffer systems are how the body maintains acid-base balance, and we're going to look at these buffer systems in a little more detail in just a bit. But so basically this is another function of electrolytes in the body. Electrolytes function by helping to maintain acid-base balance. They function as as buffers. And so if we remember what buffers are, wait, did we define what buffers are yet? Maybe we're doing it actually in this video. But buffers are weak acids or weak bases that help to maintain the proper pH within a fluid. Okay, so there are 3 buffer systems we're going to look at, but that's the second function of electrolytes, is they play that role of buffers and help to maintain the acid-base balance within the fluids of the body. Third function of electrolytes in the body is to carry electrical current. So we know that electricity is the movement of charged particles. If we didn't know that, we know that now. Let's pretend we've known that our whole lives. Electricity is the movement of charged particles. And so what are the charged particles within the body? In the body fluids, the charged particles are the ions. And so if we move ions from one place to another, in a very specific and distinct way, we essentially are making an electrical current flow. And I think we've mentioned already that electrical current that flows along the membrane of certain types of cells is all about how the nervous system works. It's all about how muscles contract. So that flow of electricity that we use electrolytes for is key for major functions of the body. And so what we're looking at here is called the sodium-potassium exchange pump, or sometimes just the sodium-potassium pump. So we know in the name, there are 2 electrolytes here named in the name of this pump, the sodium-potassium pump. So any membrane that carries an electrical current, like nerve cells or muscle cells, have embedded in the membrane this pump, which is a protein. But what it does is what it has the ability to do is pump sodium ions out of the cell and in exchange pump potassium ions into the cell. And so it does that, it runs that process, and it by doing so is moving these ions against the concentration gradient. So the concentration gradient is the concentration of those ions in a fluid. So if the concentration of, say, sodium outside the cell is already higher than the sodium inside the cell, but the cell wants to pump more sodium out, then it's pumping it against the concentration gradient. In other words, it's pumping it to where there already is a high concentration, which just means that it takes some energy to do that, and that's okay, because that's what these cells are set up to do. And that's what the sodium-potassium pump does. So any cell in the body that carries an electrical current will have this sodium-potassium pump in it. And these ions, specifically sodium and potassium, are ones that are used in that process of creating and carrying an electrical current. So that is function number 3 of electrolytes in the body. Fourth function of electrolytes in the body is electrolytes serve as cofactors. So what are cofactors? Cofactors are little parts of enzymes. So we had talked about what enzymes are. Enzymes are molecules that catalyze chemical reactions. So every chemical reaction, virtually every chemical reaction that happens in the body, we have enzymes that facilitate that reaction, that allow that reaction to occur with the use of less activation energy. And so that's just helpful in the body. So we don't have to have such high concentration of things. We don't have to have high temperature in order for the reactions that we want to occur to happen at the rate that we want them to. We have enzymes instead that facilitate. But we don't want all of our enzymes working all of the time, full steam, because we don't want all of our reactions constantly happening. So enzymes go in and out of being able to function. And the way that we regulate that is with what are called cofactors. Cofactors are either a vitamin or a mineral. Any mineral that we have in our body is we would consider it an inorganic either molecule or an atom that is part of the inorganic chemistry of the body. And so those cofactors, like magnesium, for instance, is commonly used as a cofactor, but a lot of vitamins, B vitamins and stuff, and vitamins may be small small organic molecules in some cases, but in many cases, minerals are always going to be inorganic. In some cases, vitamins might be inorganic molecules too, but vitamins can be small organic molecules, but I digress. A cofactor is, let's say in this case it's a mineral, because we're talking about inorganic chemistry and we're talking about minerals in the body. So it's a mineral component to an enzyme that can kind of plug in and plug out if it needs to. So if we need to activate an enzyme, we can plug that cofactor in, and now that enzyme is up and running and is functional, and now it can interact with the substrate and facilitate whatever chemical reaction it is made to facilitate. And then when we don't need that reaction happening anymore, then that cofactor can come off of the enzyme, and now the enzyme just hangs out in an inactive state. So that's what cofactors are. They activate enzymes or make enzymes complete so that they can function. Okay, so that was our fourth function of electrolytes in the body. Now, let's see. Let's go back to our notes here. Okay, so one of our four functions was to maintain acid-base balance. And so we're going to take a closer look at that function of maintaining acid-base balance in the body. So we looked at four functions of electrolytes in the body. One of those four is buffers, or maintaining the acid-base balance in the body. So let's take a look at how that happens. So there are three ways that the body maintains pH in its fluids. It uses buffer systems, it uses exhalation, and it uses the production of urine. And so we'll touch on each of those three ways. And we'll just touch briefly on the exhalation and the urine production. But buffer systems we'll look at a little bit more, because the buffer systems are, I mean, really, honestly, buffer systems are at work in all three of these. But buffer systems are also at work in other fluids of the body, in other places, not just the lungs and the kidneys. So of these three ways that the body has of maintaining pH, buffer systems are, like I said, they're active in all the fluids of the body, and there are three major buffer systems that the body uses, and so that's what we're going to take a look at today, is those three major buffer systems of the body. So this is where we define buffers. So we know that strong acids contribute hydrogen ions to a solution, and if it's a strong acid, it contributes a lot of hydrogen ions really rapidly. And we know that hydrogen ions are reactive and change the molecules in the solution that they're in. We also know that bases, we can either think of bases as contributing hydroxide ions to a solution, which they can do that, or we can think of them as pulling hydrogen ions out of a solution. So they're kind of making like a negative amount of hydrogen ions in the solution, in a sense. So either way, that would be either way of thinking of a base would be accurate. Or could be accurate in different situations. And so strong acids and strong bases are going to have a strong effect on the fluid that they get put into, because hydrogen ions and hydroxide ions are both very, very reactive, and they will make changes to those molecules that they're around. So what does that have to do with buffers? What buffers are not strong acids and strong bases, but we use them to make strong acids and strong bases not so strong. So buffers are weak acids or weak bases that basically can absorb the effect of a strong acid or a strong base, and by doing so neutralize or minimize the effect that strong acid or strong base has on the fluid that it's in. So we have a number of buffer systems that we have three buffer systems, three main ones that we use in the body that we're going to look at. But they're all going to essentially work the same way. What we're going to see in a buffer system is either a weak acid or a weak base, or we'll see both a weak acid and a weak base, because buffer systems can often run in both directions. And we'll see those having an effect on the hydrogen ions in the solution. So let's take a look at our first one. Our first one is the protein buffer system. Oh, this is just a review of the places where we find these buffer systems. So the first one we're looking at is the protein buffer system. So that's the one right in the middle of this chart. The protein buffer system is hard at work constantly inside cells. So it's in the cytosol. It's also in our extracellular fluid. So this, basically wherever proteins are found, which is virtually everywhere in the body, we're going to see protein buffer systems at work. So let's take a look at how these guys work. So remember, first of all, that proteins are made out of amino acids. So we string a bunch of amino acids together, give them a nice shape, like turn them into some sort of a sculpture, and then it's a functional protein. But the building blocks of that sculpture that has 3 dimensions and all these interesting facets to it and stuff, are amino acids. And so there can be hundreds or thousands of amino acids that make up a protein. Every single one of those amino acids has this same structure to it. So what we're looking at here is this central carbon that we see, that is the black carbon in the middle. That's kind of the central aspect of an amino acid. So remember that carbon can form 4 covalent bonds, and so that's what we see here. This carbon, this black letter C in the middle, is that central carbon, and it has 4 covalent bonds, one with the hydrogen above it, one with a nitrogen to its left, one with a carbon to its right, and one to a letter R that's down below. So that letter R that's down below is what's called a side chain. We have 20 different amino acids in our body to make up all of our different proteins. And on each of our different amino acids, there's a different side chain. So that side chain on an amino acid is what differs one amino acid from another. Okay, so actually the side chain has nothing at all to do with amino acids or proteins acting as buffers. So what does have to do with them acting as buffers are both the things on the right and on the left of that central carbon. So on the left, I think the first thing we have here is on the left, we have an amino group. So we have nitrogen with 2 hydrogens. So let's see here. Oh, no, first we're looking at the carboxyl group. Okay, let me go back here. So the carboxyl group is the one on the right, okay? And this is like, amino acids are like floating in this 3 dimension. It doesn't have to be on the right and on the left. I'm just saying, so we know where we're looking on the diagram. So on the right here we have the carboxyl group, which is a carbon double bonded to an oxygen, and then a single covalent bond to another oxygen that has a hydrogen bonded to it. That's the carboxyl group. So the carboxyl group on an amino acid can act as an acid by releasing hydrogen ions into the solution to lower the pH when necessary. So let's take a look at how this works in this reaction. So on the left side of the arrow, at the beginning of the reaction, we see the amino acid. So unfortunately, this amino acid is oriented differently than the one we just looked at. It has the side group up on top versus on the bottom, but it's all here, right? We have the amino group on the left, we have the side group on top this time, we have the hydrogen down on the bottom, and then on the right we have the carboxyl group. So this carboxyl group can act like an acid. What does that mean? Well, we know that an acid, when we put it in water, releases hydrogens into the solution. Hydrogen ions. So if we run this reaction to the right, if we follow the arrow, what that means is our reactant is this thing on the left, which is our amino acid, and then on the right we have our products, which are that amino acid that has undergone one small change, and that one small change is, if you look at that carboxyl group, we see that it says COO with a negative plus a hydrogen ion. So what that means is a hydrogen ion has left the building of the carboxyl group and entered the solution. So by being in this solution, that carboxyl group that's part of the amino acid has donated a hydrogen to the solution, thus making that solution more acidic. So it has raised the pH of that solution. So every single amino acid in our whole entire body, in every protein, in every fluid, has the ability to do this, for the carboxyl group of the amino acid to donate hydrogen ions to the solution when necessary and lower the pH. And that's not all. Wait, there's more. Okay, so that was the carboxyl group. If we look at the other side of the amino acid, the amino group can act as a base by absorbing hydrogen ions to raise the pH when necessary. So here in this equation, we have on the left, we have an amino acid. So we can see the amino group on the left, we would see the central carbon, we see the carboxyl group on the right of that amino acid. We see the side group above and a hydrogen down below. So let's say that amino acid is in a solution that is slightly acidic. So that solution has hydrogen ions floating around in it. So that's what we see on the left side of the arrow. On the right side, that amino acid, and specifically the amino group of the amino acid, has absorbed that hydrogen ion out of the solution, but it hasn't changed really the structure or the function of the amino acid as a whole. So instead, now what we have on the right side is we still have the central carbon, we have an unchanged carboxyl group on the right, we have an unchanged side group on the top, and unchanged hydrogen on the bottom, and on the left, that amino group has added on that other hydrogen ion, and now it's NH3 and has a positive charge to it, but we've taken that hydrogen ion out of solution, thus raising the pH of the solution. So we've removed hydrogen ions out of the solution, so we can see that the amino group has acted as a base. Okay, so all of our amino acids can do that. Proteins can buffer both acids and bases because they can act, part of each amino acid can act as a weak acid, and part of each amino acid can act as a weak base. And so the amount of an amino acid, or the number of amino acids in a given solution will be able to play with that and have an effect on the pH of that solution. So again, protein buffer system works inside a cell, so inside cells, and it works in the plasma of the blood, or well, in extracellular fluid, but plasma of the blood is one of those. All right, let's look at our next buffer system. Our next buffer system here is the carbonic acid bicarbonate buffer system. And so what we see here is, let's start here on the right. This is carbonic acid. So carbonic acid is how our carbon dioxide tends to run around in the blood plasma, but it's an acid. And so we can use it as a weak acid. In other words, we can use it as a buffer. And so we can, if we run this reaction to the left, if we're starting with carbonic acid, if we need to make a solution more acidic, one of the hydrogen ions can come off of that carbonic acid, and so then what we have is the left side of the equation where we have hydrogen ions in the solution, and then what's left is what is called a bicarbonate ion, which is a weak base. And so, but we can also run the reaction in the other direction. If we need to absorb some hydrogen ions from the solution, that bicarbonate ion can do that and turn itself back into carbonic acid. So again, it's the amount of each of these things in a solution that will help to dictate the pH of the solution. So if we need, well, say a bunch of carbonic acid gets dumped into a solution, that's going to make it more acidic because inevitably some of that hydrogen is going to come off of there. But if we put bicarbonate ion into that solution to act as a weak base, it's going to absorb those hydrogen ions, and the more bicarbonate we put in and we absorb more and more hydrogen, then less of that carbonic acid is going to dissociate into the hydrogen ions and the bicarbonate separately. So that will change the pH that way by absorbing the hydrogen ions from the solution. Okay, so we can run this reaction either way to maintain the proper amount of hydrogen ions in a solution. So we also actually could look at another side of the same equation. Eventually we're going to see this whole equation all strung together. But so again, carbonic acid on the right came from carbon dioxide and water on the left. So carbon dioxide is a normal byproduct of cellular metabolism. When it combines with water, it turns into carbonic acid. So this is partly showing us where that carbonic acid came from, but also telling us where, that carbonic acid is a weak acid and can make things more acidic. So we can use it as a buffer to lower the pH of things in the body, if we need to donate hydrogen ions to a solution. But we actually can, this only has an arrow pointing one way. It should be pointing both ways because we can run this reaction both ways as well, because carbon dioxide is going to combine with water in cells. It's going to float around in the bloodstream as carbonic acid, but at the lungs it's going to dissociate again back into carbonic acid and water, and that carbon dioxide will be exhaled. Anyway, but I'm actually getting ahead of myself in my quest to finish up here. Okay, so we just looked at the carbonic acid bicarbonate buffer system. Our third buffer system we need to look at here is the phosphate buffer system. Phosphate buffer system is largely happening inside cells. So it's going to happen, it happens inside kidney cells, but not exclusively there. It happens inside all sorts of body cells. The phosphate buffer system uses, let's see, so where do we want to start here? Let's look at the dihydrogen phosphate, which is a weak acid. If we take that dihydrogen phosphate and we put it in a solution that is very alkaline, has hydroxide ions in it, that dihydrogen phosphate is going to donate one of its hydrogen ions to that hydroxide ion, basically neutralizing the hydroxide. So we know that hydroxide is as alkaline as things get. But if we can put a lot of dihydrogen phosphate in a very alkaline solution, the dihydrogen phosphate will donate a hydrogen to that hydroxide ion, turning it into water, and we know that water has a pH of 7, so it has a neutral pH. So we've gone from something that is very alkaline, in other words, a strong base, and we've mixed it with a weak acid, so that weak acid has donated its hydrogen ions to that strong base and turned it into a neutral, more neutral. We've brought it closer to neutral solution. So in the solution now we have water and a weak base rather than a strong base and a weak acid. And again, we can run this buffer system either direction. And that can raise or lower pH depending on the amount of each of the weak acid or the weak base that's in that solution. That's the phosphate buffer system. Okay, you guys might be wondering and worrying, how much of this buffer system business do I really need to be able to spout out? Nothing really. I want you to appreciate how buffer systems work. I want you to have an understanding of how they work. And let me say this too about buffer systems, that buffer systems can get thrown out of whack in the body, but it takes a lot and quite some time of imbalance to kind of use up all of the buffers we have. But that happens. Like if somebody's in ketoacidosis, a diabetic ketoacidosis, for instance, their blood will have been becoming more and more acid over a fairly long period of time. Well, like days or weeks or months. But the buffers in that meantime would be trying to keep the pH of the blood at a proper pH. So it's not until that the buffers are completely used up and the pH of the blood really begins to drop that the person is going to experience ketoacidosis, for instance. So that's one example of how buffer systems can actually get all the way used up, and but it would take a lot to get there, it'd take a lot of imbalance to get that far out of balance where the buffer systems are no longer effective. Okay, here's our little review. 3 ways in which the body maintains pH: buffer systems, exhalation, and urine. We looked at 3 buffer systems. We're just going to briefly here look at exhalation. So our normal course of cellular metabolism makes the body more acidic, and we have buffers in place to keep things from getting too acidic, but we constantly have to get rid of acids so that our we don't get too out of balance. So exhaling carbon dioxide is what the lungs do to keep us in a minute-to-minute balance of proper pH in the blood. So it actually the lungs use that carbonic acid bicarbonate buffer system. And like I mentioned before, when the cells produce carbon dioxide, that carbon dioxide is going to combine with water and create carbonic acid. The more carbonic acid that shows up in the blood, the more the blood becomes acidic. And so hydrogen ions then begin to show up in the blood. But once the blood shows up at the lungs and is ready for and there's a gas exchange ready to happen there because we're ready to exhale, the enzymes in the lungs will drive the equation to the right. So the bicarbonate ion and hydrogen ions, if they're separate, they'll come back together into carbonic acid, and that carbonic acid, and even if they don't, we might leave some hydrogen ions in the blood, and that's okay. But whatever carbonic acid is there in the blood, that reaction is going to get driven to the right, and the carbonic acid is going to separate into carbon dioxide and water, and we will exhale those waste products. So that's going to have the effect of decreasing the amount of acid in the blood. In other words, that it's going to lower our blood pH. So if we don't blow off that carbon dioxide, we keep carbonic acid in the blood, and the blood becomes more and more acidic. I mean, so you can actually just hold your breath for as long as you can and feel the effects of this. Because what you're doing when you're holding your breath is you're denying the lungs the ability to get rid of that carbon dioxide. So you're driving the pH of the blood down just a bit by allowing acid to build up in the blood. And then once you do exhale, what you notice is you're a little bit out of breath for a minute. So your heart rate is up and your breathing is up for a little bit. And what you're doing with that increased heart rate and breathing is blowing off that carbon dioxide to quickly, within a minute, you bring your blood pH right back to where it should be, but you're lowering that you're raising the blood pH by getting rid of the acid that you had let build up while you were holding your breath. Okay, so that's the lungs and their job of getting rid of CO2. The kidneys have a job of getting rid of hydrogen ions. So hydrogen ions need to constantly be eliminated from the body, because hydrogen ions, at their at the most basic level, they are acid. And so we need to get rid of that acidity to keep our body in correct pH balance. So the kidneys actually both use the phosphate buffer system and the carbonic acid buffer system. And that's happening in different fluids of the kidneys and in different cells of the kidneys to constantly be regulating the pH of the blood. So the kidneys essentially can have the ability to pull hydrogen ions out of the blood and put them into the urine. So the urine is going to be a little more acidic, but that's okay. That's its job, to take on those hydrogen ions so that we can eliminate them from the blood. All right, so the kidneys keep sort of an hourly balance, whereas the lungs keep more of a minute-to-minute balance. But if either of these systems fail, the blood is going to begin to become more acidic, and we will begin to feel the effects of that. All right, that is it for today, my friends. So we looked at the intracellular and extracellular fluids. We named them. We talked about the barriers between them. We defined electrolytes and gave 4 functions of them and examples of those. And we talked about 3 ways in which the body maintains acid-base balance, which is through buffers, through exhaling carbon dioxide, and through getting rid of hydrogen ions in the urine. So your homework here is to choose 2 of the 5 elements that are listed here, which are sodium, potassium, calcium, magnesium, and chloride. So choose 2 of those, and for the 2 that you choose, I want you to answer these 4 things. One is, what tissues of the body are these electrolytes found in? So you're going to have to do a little research and see what you can find. So what tissues of the body are these electrolytes found in? The 2 that you chose. What specific functions do they serve? So you may find specific enzymes that they're cofactors for, or specifically that they're doing a lot to conduct electricity in the body, or whatever. So tell us what specific functions the ones that you have chosen to research serve in the body, and then find out what signs and symptoms happen if somebody is deficient in those particular electrolytes, and then find what foods are abundant in each of those electrolytes that you chose. Okay, so you can type that up, send your answers to me, email them to me, and that's it for this class. So I hope this was enjoyable. Have a good time with this homework, and I'll see you in class next week. All right, out.